Hello everyone, welcome back to my Cellchat video tutorials. From today I'm going to make videos and show you how to compare cell-cell communication networks across different biological conditions. So I'm going to use the online tutorial data again. We downloaded the data set before. So let's load the data in. So in my previous videos, I used this dataset and showed you how to do cell communication network analysis using the sequencing data from the injured human skin cells. This dataset also contains sequencing data from normal human skin cells. So today we are going to create a Cellchat object for normal cells and analyze it. Then we can compare the cell-cell communication networks between normal and injured human skin cells. So the cells were labeled in the metadata as the conditions. If we have a look at how many cells are there, you can see we have 5,011 injured cells and 2,552 normal cells. So we can get the matrix data from the R data and also the metadata for this dataset. So you can see from the metadata we have 7,563 cells. So this is the total number of both injured and normal cells. So it will be the same for the matrix data. If we have a look, you can see the matrix data also has 7,563 cells. So because we analyzed the injured cells already, today we only need to analyze the normal cells, so we can set the cell used as the normal cells NL. Then we can subset the data to keep the matrix data and also the metadata for the normal cells. So we can subset the matrix data first, then metadata. So you can see now in the metadata we only have 2,552 cells. It will be the same for the matrix data. You can see it is 2,552 cells. So now our data only contains the normal cells. We can have a look at the cell labels. It will be the same as the injured data. We have 12 cell types here. So now we can create a Cellchat object for the normal cells. The object will be the data input and the metadata will be the metadata for the normal cells. We use the labels for the group by function to label all the cell types. So let's create the Cellchat object. So you can see we have a Cellchat object for the normal cells now. At the moment you can see the net P are 0 and also the database is 0. Then we can start to analyze the Cellchat object for the normal cells. So for today's demonstration, we are going to perform all the analysis. In order to quickly finish the analysis for the normal cell dataset, I will skip all the visualization steps. So from today's analysis, you can see the simplified version of cell-cell communication network analysis using the Cellchat package. So first let's add the Cellchat database into the object. Because we only select the secreted signaling pathways when we analyze the injured cell data, so we are going to select the secreted signaling pathways again from the human Cellchat database. So we can set the secreted signaling pathways as Cellchat database use. Then we add the database into our Cellchat object. We can have a look at the Cellchat object now. You can see we added the database into the object, and it only contains the secreted signaling pathways. So we have the Cellchat database in the object, then we can subset the dataset. The purpose of subset dataset is to save your computer resources, because we don't need to analyze other genes that are not presented in the database. So let's subset the data. Now we can pre-process the expression data. First, we can identify the overexpressed genes. Next, we can identify the overexpressed interactions. For the injured dataset, we projected the gene expression data onto protein-protein interactions. So we will do the same for the normal cells and project the data onto protein-protein interactions. Because this is a human dataset, we can set ppi.human. If you are using mouse dataset, you can set the ppi.mouse. So let's do the projection. So after the projection, we can calculate the communication probabilities. Because we project the gene expression data onto protein-protein interaction, so we will set the raw use as false to only use the projected data. So Cellchat uses the trimming statistical method to calculate the probabilities. So after the calculation, we can filter the communications. So we filter out the signaling pathways that show interactions below 10 cells in all the cell groups. So after the filtration, we can analyze the communication pathways. Next step is to aggregate the cell-cell communication network. So from step 9, we start the analysis that I showed you in my video tutorial 5. So first we calculate the network centrality scores. Then after that we can identify the communication patterns. So we need to use the select-k function from the AMF package to identify the best number for the communication patterns. First, we identify the best number for the communication pattern for outgoing, that means ligand. Okay, so let's zoom in. You can see from both parameters, the pattern number starts to drop sharply from pattern 2. So we can use 2 as our pattern number. So we set the n patterns as 2. Then we can identify the outgoing communication patterns. So before we do that, I want to clear the plot first. So now we can see the communication patterns. We can zoom in. You can see there are 2 patterns. One pattern is associated with fibroblast. The other pattern is associated with immune cells. On the right hand side are the signaling pathways that are associated with pattern 1 and pattern 2. Here is the outgoing patterns. Next we can identify the incoming patterns. So we can use the select key function again. So let's zoom in. You can see the best pattern number is 4 from both parameters. So we can set the incoming pattern as 4. Then we can identify the communication patterns. I want to clear the plot again. So let's see the incoming patterns. We can zoom in. So you can see we have 4 incoming patterns. Pattern 1 is associated with dendritic cell cluster 2 and ALC cell clusters. Pattern 2 is associated with fibroblast. 3 is associated with T cells and NK T cells. And 4 is associated with T cells and inflammatory T cells. So on the right hand side are the signaling pathways associated with 4 patterns. So after the communication pattern identification, we can group the signaling pathways based on their functional similarity and also the structural similarity. So let's calculate the functional similarity first. Then we can embed the calculation into the Cellchat object. After the embedding, we can group the signaling pathways into different groups according to their functional similarity. So now we can calculate the structural similarity. Then embed the calculation into the Cellchat object. After that, we can cluster the signaling pathways into different groups according to their structural similarities. So we finish all the analysis steps. So you can see this is a simplified version that is required for all the analysis. We run all the analysis steps that we did in video 1 to 5 and skipped all the visualization steps. So now we analyze the Cellchat object for the normal cells. We can save the Cellchat object as Cellchat human skin normal cells analyzed. So now we can clear the data windows. So we saved the analyzed cell object for normal cells today. In my previous video, in video 5, we saved the analyzed Cellchat object for the injured cells. So now we can load back the normal cells and also the injured cells Cellchat object into R. So you can see we have 2 cell objects in the data window. One is for the injured cells and the other one is for the normal cells. So we can make an object list to list both data sets. Then if you have a look at the names of the object, one is called NL, the other one is called LS. So the NL is the object for normal cells. So now we can merge both datasets into one Cellchat object. Let's have a look at the Cellchat object. You can see the Cellchat object contains 555 signaling genes and it has 7,563 cells. So we can have a look at the Cellchat object in more details. You can see in the latest data slot we have 2 lists. One is NL, the other one is LS. in the NL you can see the calculated probabilities, p-values, total count, each cell type, and also total weight in different cell types. It will be the same for the LS data slot, and in the NetP data slot we also have two lists, the NL and the LS. So if we look at the NL, you can see we have 42 signaling pathways. In the NL object, we analyze the centrality scores, identify the communication patterns, and also group them based on the functional and structural similarities. It will be the same for the LS data slot. In the LS data slot, we have 47 signaling pathways. We analyzed this data set in video 1 to 5. We calculated the centrality score, identify the communication patterns, and also group the signaling pathways according to their functional and structural similarities. So now we created a Cellchat object containing both analyze the normal cell chat object and the injured cell chat object. So we can use this cell chat object in my next video tutorial to compare the upregulation and the downregulation between injured and normal skin cells. So I'm going to stop here. I hope to see you in my next video. Please subscribe my channel and share my videos with your friends. Thank you and see you in my next video tutorial.